Well, good evening, everyone. Good evening. It's nice to have you here. Well, there we go. Welcome to the School of Psychology, Family, and Community at SPU. I am Michael Groh. I'm a professor of psychology and the dean of this school, and I'm here to introduce you to my colleagues and introduce you to our speaker. But I'm also very pleased to introduce you to our initiative for individuals with intellectual and developmental disabilities. This is a new initiative. This is our inaugural year, in fact. Personal touch here, I'm a developmental psychologist, and the area of developmental disabilities is a particular area of close to my heart, both as a parent and as a professional. Let me tell you just a wee bit about our initiative before we get started. Our mission is very straightforward: to enhance the lives of individuals with differing abilities and their families. And our goal is to be a complement to the very strong educational focus in the area. Our focus is on psychological and social emotional well-being. Lifespan perspective, Dr. Wilson and I are two kind of the movers and shakers of this initiative. We're both developmental psychologists, so you can clearly see that influence there. Holistic perspective, individuals as biopsychosocial and spiritual beings. And finally, we're hoping over the course of these next few years to establish a community, to facilitate a community of individuals and families, of researchers, of teachers, of students, of lay advocates who share the core values of equality, respect, self-determination, reciprocity, and compassion. We have four aims very quickly: to support the training of our graduate students. The School of Psychology, Family, and Community has a PhD in clinical psychology, also has an MS in marriage and family therapy, and postmasters in medical family therapy. Those are the most relevant to this initiative. We also have an MA and a PhD in IO, industrial organizational psychology, in addition to our undergraduate programs. To support faculty and graduate student research related to psychological and familial issues in this area of intellectual and developmental disabilities. Third, to educate the SPU campus and local communities on the challenges faced by individuals and their families. And finally, to serve as a catalyst for a national ongoing forum among professionals, paraprofessionals, and lay advocates. Tonight is the fourth in a series of 4 speakers we've had this year. The first in the fall, 2 in the winter, and tonight, Raphael Bernier will be our speaker, and Dr. Wilson will be introducing him shortly. I'll bring up that slide for our introduction. I simply wanted to make one comment in terms of a connection with Rafe. The Center for Human Development and Disability today, back when I was a young grad student, was the Child Development and Mental Retardation Center almost 40 years ago. And I was on staff there a number of years, both in the experimental education unit and the clinical training unit, so I have a particular connection with this young scholar and professor. So with that, I'm going to stand down and ask Bev to come up.
Well, I'm excited to be able to introduce Raphael Bernier. I've known Rafe since, well, I don't know the exact year, Rafe, but when I started teaching at SPU, I had a group of graduate classes, and I really wanted to expose our students to different research groups in the area, especially research about children with disabilities. And I contacted actually Rafe's supervisor at that time, Jerry Dawson, who was here at the University of Washington for years, and she said, I have this great person you have to meet who's just going to be a star in this area. And so when I contacted Rafe, we just formed this relationship. I think every year pretty much since then, he's offered to educate our students and to talk to them about the research he's doing at the University of Washington. I found him to be not only a gifted speaker, an amazing researcher, but a very compassionate and kind person, very generous with his time. So thank you very much for being here. Just a little bit about Rafe. He did a clinical internship at the University of University of California at Los Angeles in the Neuropsychiatric Institute. He earned his PhD from the University of Washington in 2007. His primary mentor, as I mentioned before, was Jerry Dawson, one of our leading international researchers and experts on autism. And I remember one of our first conversations with Rafe was that he really came to Seattle to work with Jared. He really wanted to learn about this area and help individuals who with this diagnosis of autism spectrum disorders. And he's going to share a little bit of that research here. I could list the hundreds of publications and studies and grants that he has, but I think some of that expertise will come across in his talk to us tonight. So thank you, Ray, for coming.
Thank you. Appreciate it. It's a real pleasure to be here this evening. It's such a glorious day. It's kind of nice to cap it off with a good opportunity to talk about science, talk about autism. So what I'll be doing this evening is chatting a little bit about the genetics of autism and what we know about autism in terms of etiology and the causes of autism. So what we'll do tonight is, first we'll make sure I know how to use this thing.
Other one, yeah.
Other one. There, you got that. So, God, it's great. First I'll start with just by highlighting there's no conflicts of interest in terms of my presentation. All my funding either comes from the Simonson Foundation, Autism Speaks from National Institute of Health, or money from my book, but nothing else from any private pharmaceutical companies. I just like to make sure that quick disclosure for everyone. I'll be talking about research that I've completed as part of funding from these three sources. And so what we'll be talking about tonight is, first of all, why do we think genes or why do we think that genetics are involved in autism? We'll start with that. Talk a little bit about why is that even of interest. And then kind of turn to talk about, well, what have we learned about the genetics of autism? And I want to put that in a historical context for you, so we kind of follow a timeline of what we've learned in that context. And then, with that background, pull together and say, well, what do we currently know about autism in terms of the genetics of autism, and what are the next steps? Where are we going from here? That's the plan for this evening. So let's start with, firstly, just real briefly, what do we know about autism in general? I just want to highlight a couple quick items. First is, we know that autism is a neurobiological disorder. That is a disorder that's based in the brain. It's characterized by primarily impairments in social communication and the presence of restricted or repetitive interest behaviors. We know that. We know that autism affects boys about 3 or 4 more times than girls. We know that it does not discriminate in any way. It affects individuals in all social class levels across all racial groups. And here's kind of an important point. Autism might be better termed autisms, not autism. What we're talking about, realistically, is probably about 500, 600 different things that we all call autism. Maybe 1,000, maybe 5,000. We're not really sure yet. But we lump in a bunch of behaviors. We lump that in as autism, but really it might be more accurate to call these autisms. And I'll talk a little bit about that as we go on. We do know that behaviorally based interventions are effective at improving outcomes for individuals with autism. And we know at this time genetic or biological tests used to diagnose autism. Diagnose autism simply based on observation of the presence or absence of behaviors in social communication and doing restricted or repetitive behaviors. So I want to just lay that groundwork out first before we move in and talk about more the genetics. But lastly, we do know that autism has a strong genetic component, and here's why. So back in the 50s and 60s, certainly the idea about what might cause autism was sadly linked to a lot of parental blame. It wasn't until the 70s that we're actually going to move away from that and actually think, well, is there maybe a biological cause to autism? And so the way this question was first addressed was back by a former mentor of mine, Susan Polstein, who was a child psychiatrist who was doing some residency and fellowship work over in the UK with Michael Rutter, another child psychiatrist. And what she did, what she and Michael Rutter did, said, let's look at twins. This is kind of the technology we have in terms of understanding genetics. We know that monozygotic twins or identical twins share almost 100% of their DNA. Whereas fraternal twins or dizygotic twins share about half of their DNA. So what we can look at is we can look at the concordance rates, that is, the rates in which twinning in twin B both have autism, and compare those rates for monozygotic or identical twins to fraternal twins, thus giving some insight into how much of a role genetics plays. So in that first study, that was then followed up about 20 years later, they looked at concordance rates for monozygotic twins, identical twins, and fraternal twins, and how often both twins had a diagnosis of autistic disorder. And what they found was that these identical twins, 60% of these twins, these identical twins, both twins had autistic disorder, whereas in fraternal twins it's about 5%. I thought, this is strong evidence. We're finally able to sort of put the nail in the coffin on this whole autism is caused by parents sort of idea. That was really a holdover from, I think, Freud for this, sort of a holdover from Freud's ideas about the causation of childhood disorders. What was interesting was, in subsequent work, if you also then included concordance rates for kiddos on the autism spectrum, meaning also kiddos with a diagnosis of Asperger's syndrome or pervasive developmental disorder not otherwise specified, then those concordance rates should have been higher. And if you include kiddos with social or language impairments that don't quite meet criteria for autism, then that number, concordance rate skyrockets to about 90% in identical twins. Really suggesting that there's a strong parallel component to autism. So this is going on in the 70s and first studied in the 70s, followed up in the 90s, and there's actually been kind of a resurgence of interest in twin work in autism, which I'll actually talk about later. So that's the first piece of evidence that suggests, wow, there's something going on in terms of genetics of autism. The second bit of evidence comes from this other finding, another observation, which is termed the broader autism phenotype. And what this is the observation that family members, first-degree family members, show interesting qualities that are similar to autism, but certainly not to the point that it's challenging in autism. But behaviors that are similar to autism, again, that don't quite merit any concern, but are just interesting. So an example would be this. So one of the things we see in autism is differences in social interest or social motivation. And so on the autism end, we might see a kiddo who is really not that interested in engaging in other kids, or maybe doesn't have the skill to engage with other kids. You might see that. But this is, again, a trait that we see in all people. There's a huge range in terms of people that really love to be with people and other people that don't really like to be with other people. So what's been noted is that many family members, not all certainly, but many family members might show qualities like this that might fly on the side of not being quite as interested in being with other folks. Again, not causing any challenges, but it's an observation. What this suggests is that maybe the shared genes are resulting in the outcome of autism. So, a second bit of evidence. The third piece of evidence is the recurrence risk in siblings. So, what we've noted in a variety of epidemiological studies is that if you have a child with autism, the likelihood of having a second child with autism is about 5% or so. Although recent estimates are suggesting it might be a little bit higher. But currently this is suggesting about 5%. Now if you have two children with autism and have a third child, that risk comes up to about 20% or so, 20 or 30% of that third child having autism. Again, really suggesting that there's a strong genetic component. Given all this evidence, what it turns out is actually about 20% of all cases of autism we can actually identify what their genetic component is. Talk about what some of those components are and how we've been able to do that. So, twin studies, the observation of this broader autism phenotype, traits that are similar to autism, but not necessarily autism-like, and this increased recurrence risk suggests that there's something going on in terms of the genetics, genetic component of autism. So, great. So we find out. What's the point? Why do we care? What can we do about this? So, there's three reasons why we might want to identify the autism susceptible genes or genes that might be involved in autism. The first is that it can help aid in early diagnosis. It might help us identify the mechanism, and that will allow us to develop new methods for intervention and prevention. So, to kind of tell the story a little bit. Right now, on average, throughout the country, the average age at which parents first notice that something is not quite right with their child, who later goes on to develop autism, is between 15 and 19 months of age, on average about 18 months of age. The average age of diagnosis across the country is up to about 3.5 and 4 years of age. It's a pretty long period of time when parents say, wow, there's something not quite right. We need to go talk to the physician, or talk to the pediatrician, to actually go, and actually getting a diagnosis of autism. So, if we think about development, if we see that development is starting to go awry, and we're not able to diagnose that, we're using this period of time between 18 months and 3 and 4 to actually just do that detect and diagnose process. Development is already pretty far off the norm before we can actually intervene. So that's kind of where things are at right now, the state of the science is now. Parents make an observation, takes a while, often it's met by the physician saying, well, let's just wait a little longer, I'm not really sure, let's wait a little longer, let's wait a little longer. Oh, we should do something about this. Let's get on a wait list, wait list. Our development is heading down a different track. So, by identifying autism and susceptibility genes, it might be that we can detect right at infancy. Right at the get-go, there's an increased risk perhaps for developing autism. We can then intervene right at that point, 3, 4 months, 5 months, 6 months of age, and then ideally bring that development more in line. So, the idea is to start with a genetic test, aid in early detection, can intervene earlier, and ideally have a better outcome. We do know that the earlier we can intervene, when the brain is more plastic, the better the outcome. So that's one reason. And the other reason is to help us understand, well, the mechanism, what is actually causing autism. Because what genes do, genes are code for proteins. Proteins are really far away from behavior. So there's a lot of steps in between there. We need to understand what all those different steps are. So genes code for proteins. These proteins develop molecules, code for molecules, which then can lead to developing brain structure and how the brain functions. By understanding that mechanism better, we can intervene in a variety of areas. We can intervene in any number of these areas, primarily through pharmacological intervention. That's kind of where we're at right now. Hopefully, again, improve outcome. That's the goal. So, we know that autism has a strong genetic component. Clearly, there's a reason for trying to understand this better. It can help us detect earlier and intervene earlier. So, what do we actually then know? What has research told us? So, the story starts back in the 70s, as I mentioned, with twin studies. As we mentioned. And the second, oops, whoa, holy cow, we're on fire. That's great. So, and thank you very much for coming. That was great. Let's just jump back to, let's see if I can find. Yeah. So, great. All right. So twin studies was the technology of the day in the 70s. As we moved into the 80s, we developed some new technologies. By we, I mean, well, the field. I was playing kickball at Pine Grove Elementary School in the 80s, so really by we, I mean the field. We were to use, basically the technology was using powerful microscopes to extract the chromosomes, extract DNA, look at the chromosomes, those structures that contain our DNA, and look under powerful microscopes and see if there's anything rearranged in the chromosomes. Are there little chunks of the chromosomes that are missing? Are there chunks that are duplicated or rearranged in some way? Are those structures that hold the DNA, those instructions for our proteins? Has there been disruptions to that? We see that through microscopes. So back in the 80s, that was the technology of the day, and actually, it turns out that there are a large numbers of cases you can actually identify that there's been a change, a structural change in the chromosome. There's been a piece that's missing, that's been duplicated. And when you look at that, that accounts for about 6 to 7% of all cases of autism. You can identify that perhaps as a duplication of a very particular region of the chromosome. For example, one of the most common findings is a duplication or deletion at 15q11 to 15q13. What that means really is, let's say, for example, this is a cartoon of a chromosome. We have our centromere here, where the arms of the chromosome meet. Just by definition, a short arm that's labeled with a P stands for petite. Long arm identified with Q, and these little values, 1, and 1, 3, are just have to do with, in reference to the banding. This banding we see as a result of STEAM. So, not very interesting details, but things I just share. So, about 1 to 2% of all cases of autism are caused by a duplication at this region. A couple other regions that have been identified are deletions on chromosome 22, chromosome 2, and chromosome 16. So, this is exciting in terms of, we could say, wow, well, this, we know what is, seems to be playing a causative role in your child's autism, but we don't know why. So the regions that are missing, when you look under a microscope, these are pretty broad chunks of genes. There's lots of different genes in there. So many genes in there that it's hard to understand, well, what's the mechanism? It doesn't really help us in that way. It helps us identify what might be going on, but it doesn't really get us closer to what the mechanism is. So we moved on, as a field, moved on with our technology into the 90s, which is when I got involved, so I can actually say we now, we started in the 90s. The technology was available was using linkage studies and association studies. And what that means is, real briefly, is by identifying families where there's been more than one child diagnosed with autism. By collecting a blood sample, what you can do is kind of a complicated statistical process, but basically you can identify markers that both children with the disorder, which markers segregate with both children with the disorder. And then identify potential hotspots in both children's DNA, suggesting that there's something going on there, there's some genes located in this region that both children share. Provide some insight as to what genes may be involved. And so a study that we conducted, study we conducted back in the 90s, we worked with a large number of families on the East Coast where more than one child had been diagnosed with autism. We met with families, spent a lot of time getting to know the families, gathering information from everyone in the family, as well as a blood sample. And then we went to see, well, are there regions throughout the genome that seem to be hot spots where there's potentially a gene that might be related to autism? Is there a spot that's located? And what we found, we were very excited about this finding in the late 90s, was that there's a region in chromosome 13. What this little goofy graph is basically, it just shows, this is sort of a side view of a chromosome, and as you move down the length of the chromosome, these are lot scores, the likelihood there's a gene located at that spot. Higher the lot score, the more likelihood that there's a gene located there. This particular region of chromosome 13 seems to be a hot spot that maybe there's some genes located there that could indicate an autism, somewhere in chromosome 7. We were so excited by this finding, and so a little side note for everyone, that we're thinking, hey, we're making some headway in autism. I actually decided to take a little break from my career in academia and joined the Peace Corps with my wife, and so we took off for a couple of years. And it was like totally different. I remember thinking to myself as I'm sitting in the Solomon Islands, where I was in Peace Corps, thinking to myself, holy cow, they're going to find the genes for autism, I'm going to miss it, what am I doing, it's such a mistake. I sort of chuckled out my naivete, because wow, we're so far away from that, but there was a lot of enthusiasm at the time, I only realized that we could actually sort of find this right now. So, remember the fear that I was going to miss out, but sort of laugh. So at any rate, we're very excited by this finding of linkage of chromosome 13 and chromosome 7. But here's the tricky part. Jump ahead and talk about the logic behind it in a moment. The tricky part, the point of this slide, is to show that there have been a lot of these hot spots that have been identified in a number of studies since our study back in 1999. This just lists off a whole bunch of regions that have been implicated. Some have been reported in a couple studies, but there's been lots of folks that have reported hotspots in other regions that we haven't been able to replicate. Gosh, suggests that we're not really doing this the right way. Let me back up just a couple moments just to explain what the logic is behind this sort of model. So, a little pedigree of a family we had seen typically in our study that we conducted back east. Mom and dad, 2 children with autism, they also have a 3rd child who does not have autism. The logic behind this linkage approach is that there are multiple genes working together to confer this autism risk. So, an example here. We have a dad and mom, each have just a little side note on genetics. At each gene location, we're going to have 2 copies of the gene. We call back to biology in high school. Sometimes those genes are the same. Sometimes they're different. Same alleles at the same location. Sometimes they're different alleles. Sometimes they're dominant. Sometimes they're recessive. that's the sort of irrelevant. The point here is that you have 2 copies of each gene. In this example, in theory, that maybe dad had 3 genes that might confer autism risk at location B, A, B, and C. Mom might have 2 genes that confer risk at location D and E. Each child's going to get 1 copy from each parent. It just so happens that these first two kiddos, we got those 3 genes from dad, the 2 genes from mom. Little Joey over there on the right, he got 1 of those copies from dad and 1 from mom, but not enough to confer risk. The idea here is that when you get the right combination of these multiple genes, that's going to confer that autism risk. If you get just a couple, what we might see again that broad autism phenotype we've been talking about. That was the theory. We've got multiple genes working together that in the right combination result in autism. So the next thing, the next type of technology that came along as we've gotten bigger and faster and fancier with our computing is the notion of an association study. And this is the paper that we just published a couple years ago, the Time picked up as one of the top 10 achievements of the year, which is kind of exciting. I think that was a little bit of hype, just coming as one of the authors, but that's okay. And here's why. So what was exciting about the study is we worked, a series of us worked together to pull together a very large sample of families where there was at least 1 child diagnosed with autism. And we took all that information, scanned it, and looked throughout everywhere in the genome and said, hey, are there any places that seem to be more likely that there might be some risk associated with it? And this is a very complicated slide that is not all that important, but really here is just to suggest there's all different chromosomes in different colors on the top, and that high link is basically says what is the likelihood there might be something going on there on chromosome 5. This expands out chromosome 5 right here. This is the region on chromosome 5 we see likely there's something going on, and this just outlines the different genes and likelihood of which region within that section. What we found from this study, the take-home was that there seems to be a region on chromosome 5 that is strongly indicative of having a gene or genes related to autism. We found this in our primary sample and then replicated this in the second sample. The genes that are located there are 2 specific types of genes called cadherins, and what these genes do is they help brain cells connect and bind. So the idea is that maybe what's going on is there's disruptions in the way cells communicate in the brain, and that might be involved in autism. The challenge with this type of study is that we haven't been able to replicate it again. So it's a type of example where we have one exciting study that says, hey, we've got something going on here, and no one else can seem to find that same thing. That's one of the real problems with where we're at in terms of our understanding of using that type of technology, using this multiplex family study. So as I mentioned, a lot of regions that have been implicated, very few that have been replicated. The idea is, if you identify a spot that might be where there might be a gene located, then we can actually go explore what that gene is and what it does, and maybe that might help us understand the mechanism. So there have been a number of studies. Again, this is just a list to show the sheer numbers of candidate genes that have been implicated in autism based on these linkage and association studies. A whole slew of them, and some of these things sometimes you hear about in the media, shunt genes and orexin genes, sometimes they get kind of excited about in the media. But again, as you'll see here, there's 2 studies that support that study. There's another study that's been inconsistent, no association for this gene. There's a lot of excitement with these candidate genes, and really we haven't been able to answer any questions through this candidate gene type of work. What's interesting is it helps to sort of tell the story, but we're still pretty far away from making sense of the story. If you look at all these different genes that have been implicated in so many different studies, at least one thing is consistent, and that is many of these genes have to do with the way brain cells connect and communicate. It really suggests that connectivity is maybe playing a role. So, kind of in summary in terms of family studies. So, linkage and association studies have shown several regions that seem to be of interest in autism. But we've been having problems with replications because in order to make sense of these studies, you have to have huge numbers of families that are willing to participate. It takes time, it takes energy, it's challenging. We're certainly limited in terms of our technology. It's still expensive to run these type of analyses. And the third point, which is that there may be multiple genetic mechanisms that are working together. And this notion of heterogeneity in terms of the phenotype. Phenotype, that is the outward expression of the genotype, the behavior that we see. And if you've met one child with autism, you've met one child with autism. We know how different and unique every individual with autism is, and that heterogeneity makes things very difficult in terms of trying to make sense of the genetics. But more recently, there's been this convergence of new technology and thinking about autism in a slightly different way has helped us answer some of these questions. And that is actually a return to this notion of looking at structural changes. I mentioned things like duplications and deletions and rearrangements of the chromosome. Return to that notion, but at a much finer resolution. So back in the 70s, we were looking at microscopes, identifying big chunks that are missing. Now I can actually identify chunks that are missing on the order of actually one base pair, one little unit of DNA. So we're using much finer resolution to answer some of these questions. So what we're able to look for are these things called copy number variations. And what these copy number variations are, or CNVs as they're termed, are, again, very small deletions or very small duplications of those coding regions for genes. And what's exciting about that is sort of an interesting study was conducted back in 2007. So Jonathan Sabat and Michael Wiggler, two geneticists back east, thought to themselves, they said, hey, so we've been studying multiplex families, these families with two or more children for so long, but you know, really that's not the majority of families with autism. The majority of families with autism are families where there's one child with autism. Maybe that's going to be a good way to help us answer some of these questions. So what they did was they gathered up a number of families, what are called simplex families. These are actually families that I'd seen when I was working back east. These are families where there's no family history of autism. It's just autism seems to be kind of out of the blue for a particular family. And compared the rates of these tiny deletions and duplications throughout the genome, they compared the rates of those to these families, these multiplex families that we've been studying for a long time, as well as comparing that to typical families that don't have any evidence of autism in their family. What they found was that these simplex families, these families where autism seemed to be new, out of the blue, that the rate of these micro deletions and duplications was about 10%. In multiplex families, the rate was about 3%, and typical controls about 1%. Really suggesting that maybe there's a different mechanism at work for simplex families compared to multiplex families. So here's a very complicated slide to try to sort of share that sort of information. This is a compilation of a number of different studies looking at autism, schizophrenia, as well as asthma and typical individuals. And let me tell you what this graph shows. So on the x-axis here down here, this has to do with the size of the duplication or deletion. Okay? Far to the right, the larger the duplication. Over here, the smaller, we're talking about a duplication of 1 or 2 little base pairs. Here is the frequency with which we see a duplication or deletion, frequency with which we see a CNV. Now, typical individuals, down here in green and blue, kind of, again, hover around 1% with deletions of this size, between 100 and 500. Once we get down to these much larger chunks of DNA, sizes that we can see in the microscope, in typical individuals, that rate drops a little lower than 1%. When we look at autism, we're talking up here in the red, you see a real much higher percentage. Again, at 10% of individuals are going to see those CNVs in this sort of up to 100, 150 kilobase pairs. Those are the CNVs that we looked at in the Sabat paper. Similarly, we see a similar pattern in schizophrenia as well, with a significant drop-off in the rate, as we might expect as the larger the deletion goes. So kind of a complicated slide, but again, just want to demonstrate that idea that families where there's been one child diagnosed with autism without a family history of it, there seems to be this increased rate of these spontaneous or new deletions and duplications in the gene. So that information, along with the information we've been doing research with multiplex families, brings us to kind of where we are today in terms of what our current thinking is about the genetics of autism. So we talked about 6 to 7% of all cases we can know the cause because we can look at our microscope and identify what's going on. The other 12, 13% we can identify by the presence of these copy number variations, these microdeletions or duplications. So we know what causes autism in about 20% of all cases. That's great. We're making headway. We've moved along quite a bit since the 70s, but we're still far from having the answers. And there's two different theories now. One theory, as we talked about, is this notion that there's multiple genes working together that code for traits that are common to everyone: sociability, anxiety, interest, or that seem to be sort of range in terms of how unusual or typical they are. So a lot of variation there. The common variant theory is that with multiple genes working together, when you get the right combination, it results in autism. Whereas we have a second theory, which is called the rare variant model, and this is a suggestion that we have copy number variations, these deletions, duplications, that are rare individually, but they confer great risk. So the likelihood that if you have a deletion that is rare, it will result in autism. Two different theories on how genetics might be impacting and resulting in autism. One really based on the multiplex family work, the other based on the simplex family work. Here's what makes this so complicated, is that these two different theories are completely not mutually exclusive. So we could have both of these mechanisms happening in the same family for any number of families, really making this seem so complicated. And it's even more complicated. That's just the beginning. Talk about it gets even more complicated. So some of the complications. The first we talked about, we need to have large numbers of folks to be able to make sense of and use our technology that we have. It's been hard to work towards getting these large sample sizes. The second complication is that we have this possibly two different mechanisms working together. A common variant model and a rare variant model working together. It's very complicated to mention. Third piece is this notion of heterogeneity in phenotype. That is the outward expression. Every kiddo is so unique and so individual. How do we work backwards from that to identify what's happening in terms of etiology? And then lastly is this notion of environmental contributions. And by environment, what I mean is, the way some genes function to develop proteins that code for a structure and function of various structures, brain, bones, our skin. Some genes work to tell other genes when to turn on and off, how much they should be working, when they should be stopping. So that genetic environment provides sort of a milieu for these other genes that actually code for structure to operate. We don't know how these other genes are developing and impacting the genes that might code for brain structure and function. The other aspect of environment I'm talking about is not just the genetic environment, but also things like the uterine environment. We know that genes never act alone. The genes always act in relation to environment in some way. And so how that uterine environment will impact the expression of these genes, we don't have a sense of that yet. We can make some headway in that. We don't have a sense of that and how that might confer risk for us. So some real challenges and obstacles that are facing the field, facing all of us, facing us as parents, as scientists. So I want to kind of switch gears and say, well, what are we doing about this? And how are we making sense of this? Let's see if I can do this. There we go. Okay. There we go. Okay. So what's on the horizon? How are we addressing these complications and obstacles that I just talked about? And there are many. The first thing I just want to highlight, we've got a number of studies right now that I'm working on that are designed to actually answer some of these primary obstacles and concerns. Again, looking at working in our families to get samples that are large enough for the genetic analysis, this heterogeneity in the mechanism, heterogeneity in what we see in terms of the outward phenotype, as well as trying to understand this environment. So these are all different studies that I'm running that I'm just going to talk about some of them. I'm not going to have a chance to talk about all of them. And we'll start first with a study that I'm working on now, the Simonson Box Collection, which I think addresses this notion of getting enough information out there so that we can do our genetic analysis. So the Simonson Box Collection, what is this all about? So 5 years ago, Jim Simons, who's a hedge fund billionaire, actually, he's the 49th wealthiest person in the world according to Forbes. At any rate, well, 4 or 5 years ago, he said, friends, I have this, I've got this foundation focused on physics and math, but wow, I'm reading about autism a lot. I have a child with autism, and we really got to do something about this. I don't know what to make sense of it. So we pulled together all these Nobel Prize winners in all different aspects of science, genetics, neuroscience, physics, said, hey, let's talk with a couple people who are experts in autism. One of them was Jerry Huss, my old advisor, and another professor professor from Yale, pulled them together and said, talk to these Nobel Prize winners, tell them what's going on in autism, and maybe they have some ideas for you. So it's the first time we had gathered so many Nobel Prize winners in science to sort of answer this question about autism, and it really infused some new life into thinking about autism. So that generated a lot of excitement. What came out of that think tank was, wow, how can we make sense of this if we don't know what's causing autism? How can we make sense of it if we don't know what the genetics of autism are? We've got all these studies. Wow, people are kind of doing great work, but they're not working together. Let's make sense of this. So Jim says, well, I want to do something about this. I've really got this mission. Chat with his friend Michael Wigler, who had just published that paper by Jonathan Sabat I mentioned, just working on it. That was a paper looking at the simplex family, simplex work. And said, Michael, we got to do something about autism. And Michael said, well, I can tell you what we can do. I can tell you actually how we can answer this question. Here's what we need. We need about 2,000 families, simplex families, and we need a boatload of cash. And Jim said, well, okay, I can help you with the cash. I can't help you with the families. Well, let's do something about this. What Jim then did was pull together 12 universities throughout the country, 11 in the country and then one in Canada as well, so 12 throughout North America, and said, hey, you guys at UW, you guys are doing some great work in terms of genetics. And you guys are doing some great work. UCLA, yeah, that's great. Gosh, can't you guys work together for a second? And maybe if I give you some good money, maybe that'll help move this thing along. And so we all said, you know, that's a really good point. We do think we're doing things really the best way up here at UW, but gosh, maybe if we all work together, we could actually make some sense of this. All 12 universities, we said, let's actually work together for the first time. We made sure that when we're going to collect information, we're collecting exactly the same type of information. Because for years we've been doing this at UW, but we've been doing it the way we do it. We've done it over there at Yale, they're doing it the way they're doing it. So we said, what are we going to do? We sat down, agreed that we would all do everything exactly the same way. We videotaped each other while we're all doing this to make sure we were really doing things exactly the same way. We sat down for an evaluation. We made sure that when I was doing it here, I was doing it exactly the same way and doing it at UCLA and what have you. It's really the first time ever that we said, we can really do this. We can actually work together, wow, novel, and actually try to answer some questions, because now, the first time, we can actually get enough families and make sense of this. We've been working on, we just finished up actually data collection, ended up with actually close to 3,000 families working on this in 4 years. It was the actually largest collection, largest collaboration in the genetics of autism work that's ever been done. And we actually, for the first time, have a really wonderful network of families and researchers all working together. So we're really kind of excited. We're about a couple weeks away from being able to actually talk about the genetics finding. I can't, embargoed, so we're not actually allowed to talk about that, but soon. But I can talk about some other findings that were pretty exciting from this first assessment. The things I can leak out are that we, again, with this sort of group, replicating this notion that there seems to be a different mechanism for families where there's no family history of autism. It seems that we're seeing increased rates of spontaneous new mutations in the genome, basically deletions, duplications that are new, that aren't being inherited, that are actually happening somewhere between mom and dad getting together and kiddo. Somewhere along the line, we're seeing a mutation there. Suggests there's a different sort of mechanism in terms of genetics. Highlights the notion of how environment is playing a role as well. The other thing I'll tell you about is another interesting finding from this collaboration. So, as I mentioned, we were all doing everything exactly the same way, doing everything exactly the same way to try to make sense of what we're seeing. One of the ways we've been doing genetics research and assessment of autism over the years is we use a couple of standardized measures. One is called the Autism Diagnostic Interview, one is the Autism Diagnostic Observation Schedule. They're standardized instruments, and taken together, they're sort of the gold standard in diagnostic evaluation. So any quality clinical evaluation you might see at a university or like Seattle Children's or UW, you'll see that sort of gold standard evaluation. But there's another way of thinking about autism diagnosis, right? So the DSM, Diagnostic and Statistical Manual, is that manual that is the defines psychiatric disorders for anyone working in the field. Any clinician should have the DSM on their shelf. And that's kind of what they use to diagnose in a clinical setting. So for our study, we had all these measures we were administering everything exactly the same way. But because we were supposed to be the experts and expert clinicians throughout the country, we're certainly supposed to be the best at diagnosing autism anywhere, we didn't really specifically go into how we're going to operationalize the DSM. We just said, great, you're a clinician with a license, you've been doing this for 20 years, you know how to use the DSM to make a diagnosis for a kid with autism and decide, well, does this kid will meet autistic disorder criteria, criteria for Asperger's syndrome, or criteria for PD-NOS. These are the three diagnostic categories within the autism spectrum. So, when we collected all our data, we found that we were all seeing the same types of kids. So what these complicated graphs show is really just, there's a number of different measures we used, assessment tools we used, and the information was in different sites across the bottom. What you can sort of see here is that we're basically seeing the same type of children in terms of there was no site that saw kiddos who seemed to be more impaired cognitively, no site that seemed to see a lot more kids who didn't have language, no sites with kids who had a significant amount of seizure history. Really the same type of presentation, the same types of kids. Really over and over across all the measures. We're seeing the same types of kids. Here's where things got interesting. So in theory we all have the same data, we're seeing the same types of kids. But when you look diagnostically, we see a very different picture by site. So by that I mean, here we have again sites across the bottom, this is just 11 to 12. And this is the of those kiddos at that site, which kiddo was diagnosed either with autistic disorder, Asperger's syndrome, or PD-NOS, preexisting developmental disorder. So as an example, site 1, almost 70% of their children were diagnosed with autistic disorder, and the remainder were diagnosed with PD-NOS. There were no children seen at that site diagnosed with Asperger's syndrome. Over here we saw a sort of more representative split. This site 5, everyone was diagnosed with autistic disorder, no diagnosis of Asperger's, PD-NOS, and on and on and on. These are kind of the same kids, the same data. So in theory, you would see the same rates of these different subtypes across site. We really didn't see them. And again, we're actually supposed to be the best in the country at doing this. And how is it that we can't agree when given the same data, the same information, the same type of children? How can we agree on this? It really suggests that maybe the subtypes that we have right now in the DSM may not make the most sense. Certainly, I don't seem to be following along any pathways, patterns of presentation phenotypically because everyone's looking the same. And maybe this isn't the right way to sort of demarcate spectrum disorders. So we're very sort of, I guess, somewhat demoralized by this finding, but we thought, well, let's make some sense of it. And so as it turns out, largely as a result of this information, the DSM-5 is actually going to be changing in 2013. There's goods and bads about that, but a lot of that is driven by this finding that, wow, expert clinicians around the country can't agree on how we diagnose autism. We're very good at deciding if a child is somewhere on the spectrum, but not very good at using these particular 3 subtypes that make sense of it. So, interesting finding that comes out of a project where everyone's actually working together for the first time. Kind of exciting on that end. One other little hint I'll, that's right, we'll sort of tie to this. Exciting. So, another study we have currently ongoing that's hoping that sort of answer some of these questions. Let me just jump back to kind of highlight which question we're answering. So with Simons, we're talking about families working together to work towards this sufficient sample size complication. The other notion is the challenge with heterogeneous genetics. What are the different mechanisms? We have so many different possible mechanisms. Again, of these 20% of cases with diagnosed autism, 20 cases that we know would cause autism, there's still a lot of different causes within that 20%. So trying to make sense of that heterogeneity. We have another study ongoing called our SAGE study, which is actually a really bad name. So it was late at night, and I was trying to find a abbreviation for the Human Subject Committee as sending in the application. I really wanted to get the application in, and I thought, well, SAGE sounds so nice, but study of autism genetics exploration doesn't really ring well grammatically with me, but that's okay. So just bear with me. SAGE, it sounds nice, so we went with that for the name. But what the purpose of this study was to actually try thinking about things in a very different way. So for the past 20 years, as I mentioned, we've been focusing on families with more than one child diagnosed with autism. And when we do these studies is we work with families who are very wonderfully given so much of their time and energy to come on into our centers and into our operation and give us blood and all these other challenging things. And then what we do is we have a whole bunch of families, we try to work our way backward to figure out, okay, what might be the genetic mechanism, where are the hot spots, can we figure out what's going on? As I mentioned, that hasn't worked out very well. We've taken a new approach with the SAGE study. Instead, we're saying, has your child been diagnosed with autism? Okay, are you concerned? Are you suspected? Do you suspect that your child has autism or perhaps intellectual disability or developmental delay or something? We'll come on in. What we'll do is we do a blood draw up front. We don't actually take families' time, but it takes a lot of time to come on in. We say we like a quick blood draw. We then run a very particular type of genetic analysis, the CGH array, where we look for those cognitive variations, those deletions and duplications throughout the genome. When we look for those, identify where they are for particular families, if it's found in a family, and then at that point, we can go recontact families and say, hey, here's what we actually noticed for your family. We found out that actually there seems to be a deletion or duplication that seems to be causative of your child's autism. Would you be willing to spend a little more time with us now at this point? We then go in depth and try to understand the presentation or the outcome of that specific genetic event. Different approach, hopefully more helpful for families, because then we can actually provide some meaningful feedback and information for families. What we found in terms of sort of our first initial look at taking this approach is this. So this is just looking at 350 families, 42 of whom we found it's called a pathogenic CB, or a deletion of duplication that seems to be causative of autism. We looked at those families, those 42 families, children, compared to the children meeting outward expression or outward criteria of autism. Looked at some of the characteristics, looked at IQ specifically and language use. So we have here two different graphs here. What this graph is, it shows those children who did not have a deletion or duplication, those children who had a spontaneous or new duplication or deletion, so either mom or dad had this deletion or duplication. And then we looked at these little tiny duplications and deletions. They're either inherited from mom or inherited from dad. So mom or dad either mom or dad has this duplication or deletion. And we looked at language. What we found was these that the children who had a new spontaneous event, where there was a sudden change in the genetic structure, had a much higher rate of language delay than we saw in our kiddos without this spontaneous or de novo alteration to the DNA, which was similar to the kiddos who had an inherited event. We're not sure what to how to make sense of that yet, and so that's kind of what we sort of move forward with. The second finding sort of mirrors that sort of same type of observation. In this case, we were looking at cognitive ability. What we found was that, again, compared to those kids who have a de novo or a spontaneous mutation, compared to the children who do not have a cognitive variation, we see much lower cognitive ability, so some impairment in terms of IQ. We're trying to make sense of then what's going on. Why is it if there's a new change to DNA, why does that result in more significant challenges for a child? We're trying to make sense of it. Let me jump back again for a second. Right. So another way we're trying to address this question of heterogeneity in genetics is with another new study which we're starting up, which we'll tell you a little bit about, which we call the Simon's VIP study. This is also funded by the Simons Foundation. And it's a very different approach. It's similar to the SAGE study in that instead of, again, rounding up, working with a lot of different families and trying to work our way backwards, what we're doing is we're saying, we found over the years that there seems to be a very particular cognitive variation that is relatively common in autism. So about 1% of all cases of autism are caused by deletion or duplication in a region on chromosome 16, called chromosome 16p or 1.2, which means a small arm of chromosome 16. And so we said, why don't we work with the families who have this particular deletion duplication in this region and work forward? Let's see if we can understand what the expression is for those individuals. So we're working together with folks at Harvard and at Baylor and at Emory to work with families throughout the country to have them come on in to either one of our 4 centers, and work with us longitudinally, so we can follow their children, families across time to make sense of when you have very specific genetic structure, how does that then play out in terms of your behavior and your outcome. So it's a very different way of thinking about genetics and research. As a little side note, we've now been working on this study for just a few months. We've seen 8 families. We've had a very nice experience because I think in many ways families are saying, hey, we went to our physician, pediatrician, when we thought something was not going quite right at 18 months, we waited several years, finally got a diagnosis of autism, and the pediatrician said, hey, we should probably see a medical geneticist, see if there's something going on, go down the line. Medical geneticist says, oh, yep, indeed, we've identified that there is a mutation at chromosome 16, q1.2, thank you very much. And families say, okay, great, now what? So what's nice about the study is we're able to say, well, we don't really have all the answers yet, but you know what, at least we're going to put you in contact with all the families that have the same exact type of genotype, genetic structure. So families are very happy about that community aspect of it. And so this is all mediated through our pretty nice website that provides information about 16p1.2, as well as about the research aspect of it. So actually this summer families are getting together for the first luncheon together to sort of meet and hang out and socialize. And next year we're actually, Simon's Foundation is funding a trip to potentially Disney World for all the families and that sort of thing. So it's pretty exciting. Here's what I'm most excited about it is that this is just the beginning. So we're starting with 16p1.2 and we're going to work our way through every single genotype so we can really try to provide a community for all the different families. So slowly making our way through this sort of notion of heterogeneity in genotype. So that brings us to the notion of heterogeneity in phenotype. So the variations we see in the phenotype. As I mentioned, one child with autism, one child with autism. So how can we work backwards to make sense of the genetics if everyone is so uniquely different? Well, one way is to look at different traits and see how these different traits might be related to the genetics. So I'm going to jump right along here. So we look at these. So what we're doing is we're looking at traits that might be a little bit closer to the genotype than just saying my child has autism or my child does not have autism, which is the way most of the linguistic association studies are done. So we identify traits that are related to autism, but are maybe a little bit closer to the genetics. Maybe that might help us get there. These traits are called endophenotypes. What endophenotypes are these traits that are a little bit closer than all family members might show. So we might see the presence of an endophenotype in mom or dad at maybe a reduced level that we might see with a child with autism, but it might help us give us a little more information to work our way backwards to define what genetic mechanism might be involved. So one thing we looked at is this notion of an IQ discrepancy. This is a paper that I worked on with a medical student at UW. And basically what we did is we looked at the notion of a discrepancy in terms of verbal skills and nonverbal skills in children with autism. For the past 20 years or so, 30 years, some of the research in the clinical world has suggested, wow, we work with a child with autism and it seems like, not always, but oftentimes, there seems to be a strength in nonverbal reasoning skills relative to verbal reasoning skills. There's identifying patterns and using tasks that look at nonverbal skills to be stronger in children relative to verbal skills. So we looked at this notion in a very large sample of children, and what we found was, let's see here, the purples, blue would be what we'd expect to see in terms of the numbers, because this notion of a difference in verbal and nonverbal skills that is common to everyone. There's going to be a certain number of kiddos who have nonverbal skills and greater verbal skills, some who have verbal skills but nonverbal skills. The vast majority of people tend to be pretty evenly weighted. The blue is what we'd expect in terms of the given numbers. What we found in our sample of individuals with autism, found a much higher rate of this nonverbal skill relative to verbal skills, and about the same in terms of verbal relative to nonverbal skills. Which suggests, so we see this in a very large sample, so all this clinical lore, you can see that in a large sample, maybe there's some sense to it. So then we looked at this in family members of kids with autism, and what we found was a similar sort of trend. We found that a lot of parents of kids with autism tended to have this stronger skills in the nonverbal realm. Again, not always, far from that, but oftentimes we found them following a very similar pattern we see here. So then we said, well, we can quantify this notion of IQ, or this IQ discrepancy. There's lots of issues with IQ, but we can quantify that. We use that number. We can actually plug that into our statistical models and find out where in the genotype might there be a hotspot that's related to this discrepancy. And what we found, we're very excited about, we found that on chromosome 10, that there seems to be some sort of strong linkage there. Maybe there's something going on with this phenotype that's associated with a nonverbal-verbal split. Hasn't answered any true questions yet, but it's pointing at getting us in the direction. And maybe this is a promising avenue. Again, as I've been mentioning throughout this talk, there's lots of times when you get these hot spots in the genome, and no one else has been able to replicate. So this just came out, we just published this paper. We need to replicate this for us to feel confident that indeed there's actually something really going on in chromosome 10, but it's promising. And the last thing I'll talk about, just for next remaining time before we have some questions, is another study that we're doing, again, to look at endophenotypes. This is using a different type of approach. So one approach was looking at this discrepancy between verbal and nonverbal skills. This approach is to use a very particular brain wave that we can see using EEG. It's called the neuron. And so let me tell you a little bit about what neuron is. Here's a good way to start. This is kind of like what's really interesting to me. So I always feel like I just chat a little bit with too much detail on it. People are like, "Whoa, you're killing me, stop!" So I wanted to slow down to make sure I sort of talk about this in a more friendly way. Back in the mid-90s, there were some researchers in Italy that were doing some work with monkeys. And what they would do is they would open up the monkey brains, and they would stick in electrodes into their, this is not work I do, thankfully, and they would stick these electrodes into the monkey brains and look at sort of activation of very specific neurons. This particular group was working with 2 monkeys, and they were trying to study the motor system. So they identified specific motor neurons, brain cells that fire when the monkey was performing an action, and they were recording that activity. So they had the monkeys seated on the table in front of them, they had a little raisin they were grabbing. And these little electrodes that plug right into very specific neurons, goes to an amplifier, and every time that neuron fires, the amplifier clicks, makes a little clicking sound. So you hear as the monkey grabs, click, 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 click. So they're sort of tracking the activation of these very specific neurons. So here's the end of the day. The researchers were kind of cleaning up, and but they hadn't sort of disconnected the monkey yet. And the researcher came on over, this little white lab, little gloves, and went to grab the raisin on the counter in front of the monkey, who was sitting still. And the researcher heard click, 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 click. He's like, wait a minute, the monkey's not doing anything. This is crazy. This is weird. These are motor neurons. What's happening here? So through a series of very interesting and intricate experiments, they actually identified that these motor neurons were actually firing not only when the monkey performed an action, but when the monkey saw someone else performing that same action. They termed these specific neurons mirror neurons, and it generated a lot of excitement. For a while, there was talk, certainly a lot of excitement. There's a lot of folks saying, wow, you know, what DNA was to biology, mirror neurons are to psychology. This is huge. It's a huge finding. There's a lot of excitement because people started thinking, if we have an immediate representation of what we see other people do in our own motor system, that might explain a lot of our understanding of the social world, right? If I see someone else grabbing this water bottle, and that same motor system activates in me, I don't have to think about it, I just, my same system activates, I get an immediate representation of what that person's goals, intentions are, maybe a little bit what they're feeling, because I can understand their goals, I can see what they're doing. I get a representation, I don't have to think about it. I don't have to think about it. So we're thinking, this might be the neural mechanism for empathy, for our ability to imitate, for our ability to understand others' actions and goals, and theory of mind concept. There's a lot of excitement about it. Maybe it's even provides the link for how we've developed language over time, our ability to understand metaphor. All these different things, if you start thinking about it, these are all things that seem to be impaired in individuals with autism, interpretation ability, empathy, understanding metaphors, language, theory of mind. So a few of us thought, maybe this mirror neuron system, this mirror neuron thing, maybe there's some relation to autism, given what, in theory what these neurons do. So because of that, I thought, is there a way for us to look at mirror neurons in individual autism? Certainly not open up in anyone's brains. I'm not doing that with people. So we looked at the EEG. So what EEG does, it records the electrical activity in our brain from the scalp. There's been a lot of research since the 50s that suggests there's a very specific brain wave that captures activation of neurons that fire, that activate when we're watching other people perform actions. And there's been some a lot more recent work driven by this exciting mirror neurons that the specific brain wave is really capturing activation of these mirror neurons. So I thought to myself, it seems to be a non-invasive way to look at the brain activity. And I say non-invasive because this is my daughter, and this is the only time in about 2 hours that she wasn't screaming and crying when she had this little cap on. So I sort of say non-invasive, but yeah, you tell me it's sort of invasive to have this little shower cap on your head if you don't want to have it on your head. But it's from my perspective, it's better than like an MRI scanner or some other methods of looking at what's happening in the brain. At any rate, what I decided to do is, hey, let's actually look at then this mirror neuron system using electroencephalography, using EEG, to see if we can figure out what's happening in autism. The theory would be that then we wouldn't see the same type of activation for individual autism as typical individuals if mirror neuron system was not functioning properly. So what it is, brought in a group of adults with autism. I'll start with adults because, again, as I mentioned, this can be non-invasive, but it's invasive. I want to start with adults. And what we had adults do is come on in, perform a very basic grasping action. So they just grasp this little block of wood, which we call the manipulandum, just because that was kind of a fun name. Looks like this. They grab this little block of wood, it was in a signal to our amplifier, to our data collection computers, at the same time as we're recording electrical activity in the scalp. And then they watched me grasp this little wooden block. It's actually pretty boring too, but again, good way to start with adults because adults can sort of sit through this very boring, repetitive action. And so when it's an individual at rest you get a very, it's just a model on real data, you get a very characteristic brain wave. When both observing and executing an action, we get attenuation of that rhythm. Suggests there's activation of those neurons. So what we found was when adults with autism were grasping this little block of wood, we see exactly the same type of activity we saw in our typical adults. Exactly the same. So the motor aspect of these neurons underlying this EEG brainwave functioning totally typical. When observing another person performing a very boring action, we found typical individuals show attenuation of that rhythm, but our individual autism did not show that characteristic U-wave change. Really suggesting there was maybe something that is functioning differently in this system at the observation level. The motor system seems to be intact, but it's that translating visual information and representing that in the activation of these neurons that seems to be disrupted in autism. So, in the interest of trying to figure out how we can work our way back to the genetic side of things, I thought to myself, what if we looked at this electrophysiological indicator in parents of children with autism? Might we see a similar trend? Again, because its potential role as an endophenotype, this might help us make our way back into understanding what the genetic mechanism might be. And here's what we found. This is two very complicated slides. I'll try to make them a little more easy to read. So we have two different conditions. And we're looking at the observed and exit condition, green and blue lines. There are typical parents of typical children, parents of children who have one child diagnosed autism, parents who have two children diagnosed autism. And this is the amount of activation of the mirror neuron system, essentially. What we found was, these are both moms and dads. What we found was that our typical, that we see that sort of characteristic activation of this mirror neuron system in the parents of typical children. We see very different activation of the system in parents, regardless of whether there's one child diagnosed with autism in the family or two children diagnosed with autism in the family. It's very preliminary work that suggests that maybe this might be a potential way to sort of work our way backward towards the mechanism. Another potential endophenotype, similar to this nonverbal verbal split. We're working our way there. Not there yet, but trying to think about ways to work backwards. Talk just for 2 seconds about another study we're just beginning. As I mentioned, the 70s were driven by twin studies. That was the technology that was available. There's kind of been a swing back towards understanding twins. And so I have funding from Autism Speaks right now to look at families of identical and monozygotic and dizygotic twins with autism. And so what we're doing is we're looking at the same sort of principle, this EEG mu rhythm. But this time, instead of boring children with grasping little blocks of wood and watching other grasp little blocks of wood, we're really looking at the observation of biological motion versus non-biological motion. And the reason is this. If these are mirror neurons that we're activating, if this EEG mu wave is actually reflecting mirror neuron system activation, it should only be active to neurons that perform, it should only be responsive to things that we see that are within our biological, our motor repertoire, right? These are motor neurons, so we wouldn't expect to see activation if we're watching a ball bouncing, say, for example, because generally we can't bounce like a ball, so we wouldn't see that activation. So we're comparing activation when observing non-biological movement to biological, to movement that is within our behavioral repertoire. That is this dancer. And the reason we sort of this avatar is because we're trying to eliminate a lot of the other stimuli that's out there. We want to focus on the biological movement aspect. And it's sort of fun to watch this dancer. This avatar comes from work that was done in Scotland. There's sort of crazy research that's done. So anyway, this researcher in Scotland, a psychologist who was studying, well, what are the best dance moves? That was really a good research study. So he brought a bunch of people and he had a series of avatars, which he sent me all of them. They were doing a variety of dance moves. This is supposed to be the best dancer. And so studied, well, what are the best dance moves? And I thought to myself, that is just bizarre. I wish someone paid me money to study things like that. It's weird. But anyway, very nice of him to share this sort of stimuli to look at sort of electroactivity when observing biological motion. So trying to see that maybe if we can use, if this is a potential endophenotype again, this EEG mu rhythm, how does that segregate and how does that compare in terms of autism? So certainly we're doing a lot of work. There's no way to do any of this work without families. That's the most important part of all the work that we do, without a doubt. And there's certainly a large folks, people on my clinical team, as well as my collaborators that I work with. It's certainly a huge team effort that all of us are working on. So, again, the funders provide this, fund for this sort of work. We're getting there. We're not there yet, but I'm optimistic. So, thank you very much. Yeah, sounds great. Happy to answer any questions that work for folks. Yeah.
If you believe that the incidence of autism is increasing, is there anything in the studies that would suggest a reason for that or reasons?
So that's I like your preface to if we increase in the notion, if we believe in this notion of an increase in autism. It certainly does seem to be there does seem to be some secular increase in the rate of autism. Certainly there's lots of changes that have been going on in the diagnostic criteria, the way we diagnose autism, the way we actually do as epidemiological studies to understand what the rates of autism are. Even when we control for all these sorts of things, there does seem to be some secular increase. Are we sort of in an autism epidemic? I'm not sure if we can say that based on the data, but we can certainly say that there does seem to be some measure of increase. So the question would be, if there is such an increase, is there work that I'm doing or that's been done elsewhere that's trying to answer some of the questions from the genetic side? Well, I guess what I would say that one thing I think that is interesting is this movement toward looking at these spontaneous or de novo changes in the DNA. And that really suggests again that there are things that are happening that are changing in the environment that are potentially leading to this increased rate of changes. The things that we're doing to look at that aren't very great because it's really difficult to look at all the different environmental contributions. Again, by environmental, so we're focusing on the uterine environment, the early childhood environment. But there's so many things that moms and dads are exposed to, that all of us are exposed to, and that can impact certainly our germline and how that unfolds in terms of the genetics that we sort of either pass on or that are new. But there's no good answers yet for that. The work that's being done is limited because, again, it's so complicated trying to make sense of all these different environmental contributions. My gut, sort of my off-the-gut, data-free sort of instinct about this is that we're seeing increased rates of these mutations, probably having something to do with some environmental influences that are impacting, again, sort of genetic structure very early on, and that sort of then ending up, in the course of development, playing out as autism, and that might account for some of that increased rate. That data-free, we have yet to actually answer that question. That's sort of my hunch. Maybe we're going to be going. The work that I'm doing around the environment is looking at the exposure to various plastics, moms and parents. I'm certainly not an environmental guy. I actually collaborate with some folks at Children's that focus on this sort of work. Mine is on the genetic side. But we have to understand how these different environmental contributions are impacting the genes that we know are playing a role. And again, as I mentioned, it's not just environment from outside the body, but also the way other genes are influencing the genes that are involved in structure functions, concept of epigenetics, which is really new, probably makes sense. I don't have a great answer for you.
Great talk, very interesting. Mine is a follow-up on what you just mentioned, environmental factors, particularly lifestyle factors. You know, that can greatly influence our genetic sequencing. But how would age influence it? So is there a relationship between parents, become parents, say over the age of 40, like Down syndrome, and autism?
Sure, so there's been a lot of recent work looking at increased let's start with paternal age. That was a real strong focus. There was interesting papers that suggested that the older dads increased the likelihood of having a child with autism. Causes scare. We don't have any good answers. It's not hard and fast the way it might be with trisomy 21 or Down syndrome, which we have good, clear risk factors based on age. We're not there yet with autism. It's just on the research side of things. It suggests that the older the dad, the greater the likelihood of risk. There's been some follow-up papers that suggest actually maternal age actually contributes as well. Again, data is not super great, super clear yet. It's still sort of preliminary. But how might that happen in terms of the contribution? That's a great question. We're still trying to answer that. We do know that it increases the risk, but I'm not exactly sure what. Greater likelihood of degradation of the DNA in sperm, the theory.
At UW, where can your child go? I was wondering if UW is doing this for the physiological imaging, because I'm this mom who's very passionate about how can we design learning best around what is working well. Where is UW doing this, or what universities could you have physiological imaging, because we have to move forward with designing around, and of course working therapeutically, looking at assistive technology, visual fields, what's working well. So, these kiddos, how can you best design powerful curriculum? Who is doing this? Where is this imaging going on? Is UW doing some of it? I would love to know.
Yeah, so that's a great question in terms of, so great, with all sorts of research and figuring out cause autism, but really, we got to figure out how we're
that, right,
and bridging, helping to learn.
Right.
So there are actually a number of studies looking at the impacts of learning intervention on brain activity.
Yeah.
So there's a number of studies that we're doing at UW at the Autism Center. There's a number of studies throughout the country. UC Davis, the Mind Institute, has a number of excellent studies focusing on integrating interventions that we know work, behaviorally based interventions, and the impact on that we see in brain structure and function. The work that we've done at UW is focused using EEG, the work that I'm doing, looking at this new wave, as well as imaging work using fMRI. So I'll tell you briefly about a couple of studies we've done and what that's pointed to. So the first thing is a study, we completed a study a few years back, it's a couple of years now, using what's called the early start demo model, which is a behaviorally based intervention model. We started with toddlers and 18 to 24 months of age. Utilizes intensive intervention, basically randomly assigned children to either receiving this early start never model treatment or receiving community and regular community intervention. So we provided support, but we weren't basically providing that 20 hours of intensive behavioral based intervention. We followed the kiddos a year later, and then at the end 2 years, and now we're following them 2 years late, 2 years after that. So we just saw kiddos back in my lab when they were 6. Those kiddos that received our special intervention that the best out there, as well as intervention through the community, and looked at this EEG rhythm. What we found in these kiddos that received the treatment that we've been doing, there was a lot of variability there. Some kiddos seemed to respond, some kids didn't. What we found was we actually found normalization of that mirror neuron system activation in the kiddos who received this early intensive intervention that we did, which is strongly based in imitation skills, so learning how to imitate. So that was a way we could actually look at the way the brain is functioning differently in response to intervention. It's not, we don't really understand. We can theorize about the structure, but we don't actually see structural change. We can actually see functional change. The other study that we did that's really sort of similar was we brought in a bunch of teenagers. We looked at their ability to process information from faces. We know there's a very particular part of the brain called the fusiform gyrus that activates in typical individuals when we're processing information, visual information, about which we are experts. So many people, we process the information from faces in the fusiform gyrus. Brought these individuals in, did a brain scan, fMRI scan, looked at activation in response to looking at faces. And for individuals with autism, fusiform gyrus, I did this because it's right hemisphere based, sorry. So back here. And then we did an intensive 8-week intervention on how to look at faces. Here's how we do it. Focus on the eyes, mouth quite as much, here's what you can look at. Intensive, boring, but intensive training on how to look at faces. Then we brought this family, these individuals back into the scanner, said, hey, do we change activation? We see more activation in the fusiform gyrus, this brain region. I would not say it was clear that every single kiddo that went through this training had this remarkable change in activation. We did find that several of the children we did seem to see more normal activation of the fusiform gyrus. Some kiddos, we didn't see that change, even though behaviorally they were doing better at processing information from faces. But there's another example of where we're looking at how intervention can impact brain structure and function. Again, it's not great, it's not answering all the questions, but we're trying to take baby steps to move forward and answer some of those questions. So at least that's some avenue to think about. We do know that behavioral-based interventions work. We can see that they're having an impact on structure and function.
Do you know if the new waves, are they been related to that default mode network?
So that's a great question. Yeah, so the development, there's been a lot of excitement about the default mode network, which is basically what's happening when an individual is at rest. There's been a lot of excitement about what pathways, what brain structures are activated when an individual is at rest. And there seems to be some difference in individual autism in terms of that default mode, in terms of the brain regions that are active. And we haven't actually looked at that yet using you that. In many cases, where we use EEG, we're looking at rest as a sort of a baseline or comparison state, as opposed to thinking about it as a way to look at the default mode. But there was one study that we did looking at how different brain regions, as assessed by EEG, so meaning electrical activity recorded from the back of the brain versus electrical activity in front of the brain in individuals with autism, when at rest. We found there seems to be a decreased connectivity between these different regions. We see a lot more activity in the frontal lobe, a lot more activity in the posterior lobe, but not a lot of communication between those two. So there's some work that's been looking at that. We haven't looked at that, how it relates to the new wave.